Hello, good morning, everyone. Hope you had a good weekend, and thank you for being on time, even though we had one less hour of sleep. We do have a lot of things to go through today. My plan is to finish the sensory organ unit lesson with you, and hopefully start on the digestive system. Now, I know on the calendar. It says that the test is going to be next Monday. I don't think that's going to happen. Okay, I think it's going to be too rush. We probably have to postpone it to maybe the Wednesday or something like that. Okay, I'll let you know. I'll see where we end up at today, and then I will give you a definite answer afterwards. Okay, so last class we labeled the eyeball in the study guide. So we're just gonna go through the PowerPoint slides for it. So in this picture, you can see a person has two different eye colors. That's a real thing. It's called heterochromia. Heterochromia can either be a genetic condition, okay, where the person is born with two different eye colors, or it can happen as a result, as a symptoms of other eye diseases. Um, for example, like eye trauma, glaucoma, or surgery sometimes can cause the pigment to be lost in one eye but not the other, and that gives you two different eye color. But some people are born with this, and there's no pathology associated with it. It doesn't affect your eye function. Okay, so let's take a look at what's going on with the eyeball. So let's start with the accessory organs of the eye. It's more like accessory structures. I think that's probably more appropriate. Let me just change the accessory structures of the eye. And that includes eyebrows. Okay, eyebrows, some people think about it, not sure what the function of the eyebrows are, right? It's one of those things that we have, and it's kind of weird to not have, but then in the end, we're not sure what it does, right? But it turns out the eyebrow is important to shade the eyes from the sun. Right? It's the same reason why football player put the little paint under their eyes to reduce the glare from the stadium light. So similarly, that's what our eyebrows are for. Right? If you're sweating in the summer, the sweat comes down the forehead, it hits the eyebrow. The eyebrow kind of allows it to go to the side, so it's not going to go into your eyeball directly. We also have the eyelid, which is continuous, a continuation of the skin that we have. And of course, blinking closes the eyelid, and that helps to keep the eye from drying out. Okay, it's a very exposed surface, right? So every time we blink our eyes, we cover it with a new layer of tears, and that lubricates the eye. And also, if you remember from earlier units, that the tears actually contain lysozyme, right? And these are things that's gonna help keep the surface of the eyeball sterile. We also have eyelashes, which help blocks debris from entering the eye. So all these things are kind of, you know, extra accessory structures that come with the eye, and it protects it in various ways. This is a fully labeled eyeball. We labeled most of these things already last class. Okay, so most notably, the eye has three layers, right? We have the sclera, which is the outermost layer. Beneath it is the choroid. That's where the blood vessels are going to be. And the innermost layer is the retina, which is where you will find the photoreceptors. Okay, photoreceptors are cone cells and rod cells, as I've told you, and they allow you to see different colors. That's the cone cells. And in night vision, that's the function of the rod cells. So let's take a closer look at these three layers and see what else we can learn from them. So like I said, the sclera is the most superficial layer of the eye, it's the outermost layer of the eye. It's the white part of the eye. It's actually pretty fibrous. Fibrous basically means it's very tough, okay? Like, you can stretch it, it's not gonna tear apart. It has some elasticity to it. And it provides attachment sites for various eye muscle. So if you take a look at the eyeball here, all right, this is the right eyeball, and you can see there are how many? 1, 2, 3, 4, 5 different muscles attached to the eyeball. Right? So like we have the lateral rectus, which will help you, roll the eyeball to the side, right? Like you look at the side. You have the medial rectus, which would roll it towards your nose, and then you have muscle at the top that allows you to look up, and then this one allows you to look down. And a combination of them, like for example, if the top and the medial one pull at the same time, then you would be looking in a diagonal manner, right? So those are the obvious ones. Right? It covers the up, down, left, right. Okay? But you also have what's called like an inferior oblique. You don't have to memorize the names of the muscle, okay? Anybody can want to guess what the inferior oblique does? The other four is obvious, right? Like up, down, left, right, and anything in between. But what about the inferior oblique? What do you think that one does based on its position?
Stretch it on the side.
To the side, okay, but that's what this one is for, right? This one allows you to look to the side.
Like on the corner.
Help you rotate.
That's right. This one helps you roll the eyes. That's right. So like if it pulls, it will roll your eyes like this. Okay. So rolling the eyes is important. Not for like survival reasons, I suspect, but it's as an expression, like you roll your eyes at someone. Okay, the most anterior portion of the sclera is the cornea. Right, so here in this very zoomed in picture of the eye surface, you can see the cornea is actually a dome shape, right? And that's important. I told you last class, because the dome shape, we actually say convex, right? Okay, so convex shape. Convex is something that bulges out. It's like if you think of a spoon, the side that you eat with, that's concave because it caves in, and the back of the spoon is convex because it bulges out. So the cornea is convex, and whenever things are convex, it allows light to be collected from a wide angle. Like the security mirror, right, in its convenience store, allows the store owner to have a broad view of the store, right? So the cornea, being convex, allows us to have a wide angle of vision. Okay. There was a thing a couple years ago where people, I guess, they run out of place to tattoo on the body, and they want to tattoo it on the eyeball. This is as bad as it sounds like, right? So obviously it's very delicate tissue in the eye, and some people are actually left blind after the procedure. Okay, they try to do the tattooed ink right in the sclera, right, to have like a permanent marking in the eyeball. And so after several people went blind, they're trying to ban the procedure. When Canada has already banned this, I'm not sure what the situation is at the state, but don't tattoo your eyeball. If you want your eyeball to look special, different, you can always wear a contact lens. They have contact lens for that purposes. The choroid, okay, choroid is the next layer, is the layer beneath the sclera, and that is where the blood vessels are going to be located. Anytime you have blood vessels going to a place is for nourishment. It's providing nutrients to the eye tissue, as well as removing some of the waste product. The eye, needless to say, is alive, and if it's alive, it's going to be doing cellular respiration, right? So you do have to remove some of the waste product from them. The choroid has an opening at the front, and that is what makes the pupil. Right? The pupil allows light to enter the eye. Okay. It's like, it's like your camera on the phone, and it allows the light to go in. If you shut off, if you block the pupil with your eyelids, for example, then no light can enter and you won't see anything, right? But the size of the pupil is also important. If too much light goes in, then it's overexposed. You cannot see things clearly. You also cannot focus on say the letters on a book. Okay, and if it's too small, not enough light coming in, that could also be a problem. So it just needs to be the right size for the occasion. Okay, if you're outside, it needs to be smaller because it's bright. If you're inside and it's darker, you need it to be bigger. If you're reading, it also needs to be smaller as well to help regulate the amount of light going in. Now, this is done automatically. Okay, the size of the pupil is regulated by the, if you remember from earlier lessons, from the by the ANS. Okay, so specifically the sympathetic system is going to dilate pupil. You need more light to come in when you're in a dangerous situation, right? Whereas the parasympathetic, that's going to cause you to constrict pupil. Okay, so that's the autonomic nervous system, the ANS. Right? It's automatically done. There's no conscious input, right? Like in movies, sometimes you see the doctor shining a light in the person's eye, right? That's checking for the pupillary reflex, right? Does the pupil know to shrink in response to a bright light, right? That's a way to assess if there is any kind of neurological damage in the head. So what actually changes the size of the pupil? Well, that is the function of the iris. Right? So not only does the iris gives color to your eyes, but it's actually muscle. So when the muscle contracts, it makes the pupil smaller, and when it relaxes, it makes the pupil bigger. So the color of your eye is related to the color of our skin, right? Because it's the same pigment, okay? Same pigment that determines your hair color as well, right? So the pigment is called melanin, right? The more melanin you have, the darker the pigmentation is going to be. So people with lighter skin color, they usually have blue eyes and blonde hair. And then on the other side, if you have darker skin pigment, usually we have brown eyes and black hair, right? Because again, it's the same pigment, right? And that's genetically determined.
Sir,
Yep.
what does it mean when they say the pupils are blown?
The pupils are blown. I think it just means that it's dilated, right? So it's like really big, right? So sometimes when you go for eye checkup, the doctor applies a little bit of pupil dilator. Okay, it's called atropine, and then it opens up your pupil. It's blown up so that they can peek into your eyes. They can look at like the retina, for example. They can take a picture of your retina. It's difficult to do when your pupil is small, right? Yeah. Anybody
But
had? Yeah, sorry.
you know, in the movies when they say like
The drugs,
if
right?
they
Like,
check him for a trauma, like the pupils are blown, like he's dead or
oh yeah. So like if you had a head trauma, then this did you say head trauma? Did I misheard you?
yeah,
Yeah.
yeah.
So like when that happens, the ANS, which is part of the brain, right, may be damaged by the trauma. So you no longer have this regulation of sympathetic and parasympathetic regulation. So then the eye muscle just relax. Relax is its natural state, right? So when it relaxes, the pupil gets really big, right? Yeah. Does that answer your question?
Yeah.
Yeah, so that's what I was saying, right? Like the doctor would like shine a light at the person and see if the pupil will shrink back. right? Because that's a reflex that's supposed to be in place, right? But if it's damaged, you're not going to be able to do it. So I was saying, right, like anybody had an experience with the eye doctor where they put the eye drops into the eyes, right? Maybe you know what I'm talking about.
Yes, Professor.
Yeah, so then the pupil gets big, and it's hard to drive, like, especially if it's bright outside. So they tell you to bring your sunglasses to dry, and if you notice, it's also hard to read. I remember this very distinctly because when I was in university, I had an eye doctor appointment the day before my final exam. Okay, so I went in the morning. I did my checkup, they dilated my pupil, and I could not study for the basically for the rest of the morning. Okay, all the way until evening, and it started to get better because the pupil was so dilated. I told on to my textbook, I can't read the words. Okay, no matter where I put it, I just can't see it because again, focusing on the words requires your pupil to be a little bit smaller. Okay. Drugs as well, right? Some people, if they're on certain medications, it cause their eyeballs to dilate. You might have, again, seen that in movies. Anyways, moving on, we have the sclera on the outside. The choroid is the next layer, and the innermost layer is the retina. Right? The retina contains photoreceptors. Photoreceptors. Remember, there are different types of receptors in the body to detect different things, right? We have chemoreceptors in the mouth and the nose for taste and odor. We have photoreceptors in the retina. Okay, it's at the back of the retina because if you think about the eyeball, right, this is where the photoreceptors are going to be concentrated at the posterior portion of the eye. Why? Because the light comes in from the lens, and it's only going to be able to hit the back of the eyeball, right? Like no point putting photoreceptor here or here or here. The light's never going to reach it. Okay, so they're like concentrated at the back end of the eyeball. All right. Think of this as like the projector, right? To project the light. Where are you going to put the screen? You're going to put the screen in front of the projector, right? So the retina is like the projector. No point putting it here. The light is never going to strike it. That's what I'm trying to tell you. Okay, so there are two types of photoreceptors. We have rod cells for night vision and peripheral vision. If you had me last semester, you might remember I told you about vitamin A deficiency in the biochemistry unit. People with vitamin A deficiency, they would not be able to make a molecule that is essential for the rod cells to function at night. So they have nighttime blindness, right? But if you have adequate, healthy diet, then you should be okay. And the rod cells will help you see at night, and it also helps you see the peripheral vision. I explained peripheral vision to you last time, right? I told you to focus on reading the words on the slide, and I put up my hand, even though you are aware that I have put up my hand, but you cannot make out how many fingers I was holding up without shifting your focus, right? So the thing that you're paying attention to, the thing that you're looking at, that's the central vision, okay? And that's processed by the cone cells. Everything else is your peripheral vision, okay? The rod cells. Cone cells help you distinguish colors. Now, there are three types of cone cells, R, B, and G. Okay? Red, blue, and green. It's like those old TV monitors that you might have, okay? Like if you wipe it with a wet cloth, you see the individual pixel, the red, the blue, and the green. And it's a combination of these red, green, blue that gives you all the different colors that we are able to see. Now, people get a little bit confused usually when I start explaining this to them. Okay? For example, if I asked you, what do you get when you mix blue and green together? Okay, so people they are thinking of it in terms of like painting color. Painting color behaves differently than light color. Right? So for example, if you mix, I'll talk about painting. Okay, if you mix green paint what is it? Sorry, blue paint and yellow paint, what do you get? If you mix yellow and blue, what color do you get?
Green.
Green, that's right, okay? But for light is different, okay? We actually get yellow when we mix you know, red and blue together. Right? So like if you don't believe me, red and green, I mean, sorry. Let me try to show you with this. And you can play around with this yourself as well. Let me just put like a random shape here, okay? Circle here. And I am gonna change the color here, more filled color. So you see, I know it's a little bit small. Oh, no, making the picture bigger is not gonna make it smaller. So here we have 3 colors, red, green, and blue, right? Let's just turn it all to 0. 0, 0, 0. So if there's nothing, then it's black, right? You can look at the color right here. And if you only turn on the green cone cells, right, turn it to let's make it even bright, 200, then you see green, right? You see green, obviously. But what if you have green and blue at the same time? So you turn on the green and you turn on the blue, you actually get this color, which is called cyan. Okay, I'm just gonna make it a little bit brighter for you to see. This is called cyan. Okay. What about green and red together? So I'm gonna turn the blue back down, and then I'm gonna turn on the green and the red, make them the same, right? And I'm gonna turn it up. And you get the yellow, right? Okay, so by having a combination of these colors, by having a combination of these red and green and blue cone cells, you're gonna be able to generate basically all the colors that we see. Okay, so over here, for example, I can move the cursor, right? Let's say this color, this is called magenta, right? Magenta is mostly red and blue. Like green is almost didn't turn on. Look at the number; it's very low, right? And what if you want to see orange? Well, orange is, you know, mostly red, a little bit of green, and then almost no blue, right? So by shifting the intensity of the cone cells, you are able to detect the different colors. All right, maybe we'll come back to that later. That's what they look like in your eyes. They are really small, 20 micron. And so this is the color wheel. Okay? This is the color wheel. Right? To see yellow, you need red and green. And if you crank up red more and lower the green, then you see more of like an orange color, right? As I was trying to show you. Okay, red and blue gives you magenta, and then blue and green gives you cyan. If you turn on all of them, it gives you white. So I'm gonna do an experiment with you. I will be explaining the thing as you're doing the activity. Okay, don't write anything. For this to work, you have to follow the instruction. Right? So here is a red screen. Okay? Your job is to stare at the red screen. All right, just stare at it hard. Okay, unless you're driving, then don't stare, obviously. So what you're doing right now is the red screen appears red to you because the red light is entering your eyes. So like I said, you have 3 types of cone cells: red cells, blue cells, and green cells. Because the screen is red, only the red cells are being stimulated right now. Okay, only the red cells are sending signals to your brain, to your occipital lobe. So you're perceiving this as red, 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 red. And keep staring, okay? The blue and the green are not doing anything right now because there's nothing that stimulates those cone cells. I'm gonna switch to a screen. I want you to blink a couple of times. Can you tell me what color you see? Tell me, what color do you see on this white screen?
Looks like turquoise.
Turquoise, okay. You see white, really? Some people says white. You gotta blink a couple times. Okay, blink, blink, blink. Do you not see any other color? Does it work for anybody at all?
Grayish?
Grayish?
Yeah, it worked for me.
Okay. All right. So you can try this again later. It should work. Okay.
Oh.
Otherwise, go see an eye doctor. So
Excuse me.
yeah, go ahead.
I saw a light green, after that white.
Okay. Yeah. So let me go back to the color wheel and try to explain what was done. Right? So how do you see the color white? You see the color white if red, green, and blue, all three of them are working, right? If all three cone cells are activated and sending signal to your brain, then you see the color white. So when we were staring at the red screen, only the red is working, right? Only the red is sending signal to the brain. But when you stare at the red screen for so long, it gets tired, it gets overstimulated. So when you switch the white screen, when you need all three of them, the red is like kind of tired. So it's not really sending strong signal. So even though it's a white screen, but the green and the blue is dominating, and that's why you would see this color. And some people say turquoise-ish, right? But you know, the correct name for that color is cyan. Do you guys understand that? Okay.
Yeah.
Now there is a thinking application, whatever you want to call them, question here. Let's take a look if it makes any sense. You stare at a bright red screen for 30 seconds. Right? What color will you see if the screen is switched to white? I already told you, you're gonna see cyan, right? Okay, you're gonna see cyan. Do I even have cyan here? There you go, cyan. I know this looks like blue, but it's called cyan. Okay, what if you stare at a red screen and then you switch to a green screen? What color are you going to see? What color are you going to see? If you're staring at a red screen and you switch it to a green screen afterwards and you blink your eyes a couple times, what color are you going to see? Okay. I have yellow. Anyone else?
Cyan.
Green.
Okay, I have cyan, I have green. Okay, green screen, right? Green screen. What kind of cone cells do you need to see the color green? It's not a trick question. Okay, very straightforward. What kind of cone cells do you need to see the color green?
Green
Green.
one.
Green ones, that's right. So to see green, does it involve red at all? No, it does not. So do I care if the red ones are tired from the previous red screen? No, it's not going to have an effect, right? So if you switch to a green screen, you will still see the color green. Okay? It's only going to be affected if the new color requires the red, right? But it doesn't. Right? So what you're going to see is just green. Okay. Now, what if we switch to a yellow screen? What if you stare at red and then we switch to yellow screen? What color will you see next?
Green.
Anyone else? Blessing Mohammed, they say green. Anyone else?
Maybe white first because the red would be a bit tired, so you'd see the other ones a bit
But
more.
to medicine, but to see white, I also need to have the blue, right? Okay, is
Green.
blue? Yeah. So let's see, yellow, right? The screen is yellow.
Yellow.
Yeah. What do we need to see yellow? To see yellow, we need red plus green, right? Okay, yellow is red plus green, but the red is higher from the previous screen. So what do you see? Green. Okay. All right, let's try one more just in case. What if you were staring at a blue screen, and then you switch to white? What color will you see? Just think about it first. You're staring at blue and then you switch to white. What do you see?
Yellow.
That's right. Okay. What is white? White is red plus green plus blue. The blue is higher from the blue screen. So you will see combination of red and green, which is yellow. Does anybody want to ask anything? You will have maybe one or two of these on the test.
So if you're looking at green and you turn to the white, you will be magenta.
That's right. That's right. If you're looking at magenta, yeah, if you do magenta and then you switch to white, what do you see? Last example, okay, and then we're gonna move on. Magenta to white.
Yellow?
Okay. Anyone else?
Oh, green, green.
Okay. But anyone else?
Sir, I don't get it.
Okay,
I'm a bit confused.
that's okay. So Patrice, what cone cells are needed? Okay, do you agree that we have only 3 cone cells? Red, green, and blue?
Yes,
Yeah.
yes.
So which ones do we need to see the color white?
We need all three to see
That's
white.
right. Yeah, red, green, blue. Right?
Yeah,
Now, what do we need to see magenta, Patrice?
red and blue?
That's right. So if you stare at a magenta screen, do you agree the red and the blue gets tired?
Yes.
So when you go to the white screen, the red and the blue are tired. So only green is working for a bit, right? So that's why you see green on a white screen. Is that better? You answer all the
Me-
questions along the way. So where's the
Oh my God.
where's the confusion? That's okay. You just ask me. Where's the confusion?
I don't know, it's just I've never learned colors like this before. I just know, mixing paint, that sort of color.
Right,
I'll
right.
rewatch the lecture.
Okay. Do you
And
understand why I
Google.
cross out the red and the blue though?
Yeah, because you said, what did you say?
For
The
magenta, you need red and blue, right?
red and blue.
Yeah, and
Yeah.
that's why they got tired out, and then it's not working as well when you switch to a white screen, right? So only the green one is the dominating cone cells at that moment.
Okay.
First, I have one more suggestion. As your picture,
Mhm.
what I was doing, just choose whatever picture is outside of that color. If you're saying, if you are looking at red
Mhm.
and you go to white, so go out of the cell of the, out of the red cyan.
Go to the what? The
Outside
opposite
of the
side? The
circle.
opposite
color
side?
circle. Yeah.
The opposite? Oh, okay. So like opposite of magenta is green, opposite of yellow is blue, like that. I don't know, because what if I switch from a green screen to a red screen, right? Then it's not opposite anymore, right?
Right.
Yeah. Anyways, rewatch this if you need to. Okay, so here is another one that you can try. You might have seen this before, okay? You're supposed to stare at the dot. Okay, for this to work, you really have to stare at the dot. All right. And all the stuff that are around, like the color is all wrong. Like you might not know what you're looking at. But basically, this is the opposite color of what it's supposed to look like. As you're staring at the dot, just gonna stare for a couple more seconds to really exhaust those cone cells that register these colors. Okay, to see the purple, you need to use the blue and the red, and so on and so forth. Now I'm gonna switch in a second, and when I switch to the new image, okay, keep staring at the middle and just blink a couple times. Okay, do you see the picture as a colored picture? For the first couple of seconds, you will see a colored picture, right?
Yeah.
Yeah. But then after you blink a couple of times, you realize, hey, this is a black and white photo, right? Okay. So what happens is the same thing. Right? You to register these different shades of gray, you would need a combination of red, green, and blue. But in the previous one, we were tiring out some of them at certain locations, and that's why you're seeing the opposite color. All right, don't at most there are like 2 questions on these things, all right? So like maybe don't get too worked up on them. Okay, I already told you this one. Is there at a green screen for 1 minute and then you switch to a yellow screen, what would you see? Okay. I'll just tell you the answer. Okay, you guys can figure it out later. Okay, the answer is red. Okay. Okay. Color blindness. So some form of color blindness has to do with the cone cells not really able to register certain light wavelength. And most color blindness, as you know from me last time, is X-linked, right? Yeah. So there is a test called the Ishihara test it's a quick way to see if someone has a certain kind of color blindness. This one tests for red-green color blindness. What numbers do people see here? Anybody?
74?
74, yeah. Everybody see 74? If you see seven,
74.
yeah. If you
Yeah,
see 74,
74.
if you see 74, then you don't have color blindness. Okay, you don't have red-green color blindness. If you see 21, then you have color blindness. I mean, there's nothing wrong with having color blindness. Okay, they even have special contact lenses for people to wear if they have color blindness to help correct it. But you know, it's something that is X-linked. All right. Complete color blindness is very, very rare. And you know, you would need another test for something like that. Okay, moving on. This is like a picture of inside of your eyeball. And usually it's part of your eye exam routine. At least it used to be, but then now I think they changed the coverage and you know you have to pay for it or something. So then they take a picture through the pupil, right, inside of the eye, and you can see a few familiar things. Okay, so there is there's a bright spot right here, and you can see all the blood vessels kind of radiating from that spot. That spot is called the optic nerve. And the optic nerve carries the information to the brain, obviously. And where the optic nerve exits the eye, you have no photoreceptor there. So that is where you have your blind spot. Okay, blind spot, which is also known as the optic disc. Optic disc. Okay, I forgot to highlight the other word. Okay, so there's the optic disc or blind spot. A little bit to the side of the optic nerve is an area called the macular. Oops, called the macular. The macular is the center portion of the retina that produce sharper image. Okay, so basically, whatever you're focusing on, that image is going to be projected onto the macula. Everything else is part of your peripheral vision, and there is a little pit in the middle of the macula called the fovea, and that has the highest density of cone cells to create the sharpest image. The sharpest image. Okay. So generally, the macula gives you sharp image, like HD if you would, but the fovea gives you like ultra HD, super sharp image. Okay. And it has the highest density of cone cells in there. All right, so that's what it looks like inside the eyeball. So let's try to find our blind spot. Again, depending on what device you're on, sometimes it works, sometimes it doesn't work. Okay, if you're on the phone, then you can move the phone. Okay, if you are doing this on a computer, then you have to physically move your head. Okay, so this is what you do. Right? We're gonna cover our right eye, or you can just close it. Okay, cover your right eye, and you're gonna use your left eyeball and stare at the plus sign. Okay, cover your right eye and stare at the plus. Okay, you might already be at the blind spot. If you do that, you might see the circle disappear. But if at this point, keep staring at the plus. If you still see the circle, you can move closer or you can move further away until the circle disappears. When the circle disappears, then you have found the blind spot of your left eye. Do you guys understand this? Cover your right eye, stare at the plus, and you move slowly closer to the screen or further away. Like for me, this is the right distance already. Like when I cover my right eye and I stare at the plus, the circle disappears for me already. So like That's my blind spot for the left eye. Okay? And then you can do the same for the other eye, but this time you stare at the circle and then you see when the plus disappear, right? So it disappears because the image lands on your blind spot of that eye. Okay, there's no photoreceptor there, so you don't see it. But realistically speaking, if both of your eyes are open, then we don't really have a blind spot because the blind spot of the right eye is covered by the left eye. Like the left eye would compensate for it. Okay? Whatever the right eye don't see, the left eye is going to see it, and vice versa. So with both of your eyes open, it's okay. You don't truly have a blind spot in front of you. Okay? I mean, you have blind spots behind you, obviously, but like, you know, otherwise, it's relatively okay. All right. Let's see if you guys are up for some polls. Go for it. Okay, that's pretty good participation. The cornea is part of the sclera. It's the extension of the sclera, right? 3 types of cone cells. You should know this. Mm-hmm. It's the red, green, blue, right? Red, green, blue. RGB. How about this one? This one might be a little bit tougher. I only briefly mention it. Okay, good. Most of you got it right. It is the iris. Okay, the iris. It's the color part of the eye. Not only does it make your eyes look pretty, it also controls the size of the pupil, which helps regulate the amount of light entering the eyes. Last one here. Okay, so sharpest image is the fovea. The fovea. Fovea is a little pit that is located in the macula. Macula is the center of the retina. Macula is a bigger area. That's for sharp image, but the sharpest is going to be in the fovea. All right, not bad. All right, let's move on and see what else we have to know about the eyeball. Lens and related structure. So the lens is crystalline structure located behind the iris and the pupil. Okay, so you see here, that's the cornea. There's going to be a gap here, the anterior compartment, and then that's the pupil. and the lens is right behind it. Okay. So the shape of the lens, and more specifically the curvature and the thickness of the lens, needs to be adjusted whenever we shift our focus. If we look at something further away, the lens needs to be thinner. When we look at something that's closer by, the lens needs to be thicker. Okay. And so the lens is held in place by these suspensory ligaments, right? I showed you a 3D model last class. You definitely should check that out yourself. And on the other side of the suspensory ligaments, we have what's called the ciliary body, which is a muscle. Right? So the muscle is like a ring, right? And when the muscle contracts, it pulls on each other, and it makes a smaller ring. Okay? Whereas when it relaxes, it kind of opens up. Right? So you gotta have to visualize that. I don't have an animation for you, but the key thing to know here is when we're looking at this from the front, and we can see the ligaments here shown here by the lines, and then we can see the muscle here represented by the purple thing here, okay? The purple circle. So think of it like this, right? You have all these ligaments connected to the lens, right? So when the muscle, my hand here is the muscle, when the muscle gets big, it's actually pulling on the ligaments in all the direction, right? Can you visualize that? Right? Like so. Do I have a elastic band here? Here, let me go get an elastic band. Maybe this is gonna make more sense. Just give me a second. Okay, sorry about that. Okay, so look, this is the elastic band, and the elastic band represents the ligaments. Remember, the ligaments is going to be attached to the lens and to the muscle, right? So right now the ligament is loose, and then this is tight, right? So the ligament is connected to my hand, the hand representing the muscle. So look, if the muscle is relaxed like this right now, then it's loose, right? Okay. Sorry, one more time. This is the hand is the muscle, and this is the ligament. Okay. When the muscle contracts, it gets smaller, and you can see the elastic band is loose. But when the muscle relaxes, it gets big, and it pulls on the ligament, right? And then it becomes tight. Right? So loose, contract, and then relax, tight. Right? So let me write it down for you. Actually, it's already on the screen. When the ciliary muscle relax, relax means bigger, bigger circle, I suppose. And what happens is the suspensory ligaments get pulled tight, and then the lens becomes thinner because you're pulling on the lens on all the direction, right? It makes it stretch out, and it becomes thinner. On the other hand, when the ciliary muscle contract, right, it becomes like a smaller circle. Right? So look, small circle is very loose. Right? Big circle is very tight. Right? So the suspensory ligament becomes slack, and then the lens is allowed to bulge. So like thicker. And you need it to be thicker when you're looking at nearby stuff, e.g. reading a book. You need it to be thinner when it's far away, like looking at the sign on a bus that's like coming towards you. So in other words, you need to shift, whenever you shift your focus, then you need to change the shape of the lens. And this happens automatically. This is called visual accommodation. Visual accommodation. So I know that wasn't very good explanation with the pull and the push and the tightness and stuff like that. Any questions you want to follow up with? Okay. So as we get older, the ability to do visual accommodation is gonna diminish. Okay? If you think again, look at the rubber band, right? Rubber band right now is very stretchy. You let go, it goes back to original shape, right? But if you keep on stretching it multiple times, eventually it loses its elasticity, right? Over time, it loses its elasticity, and after you stretch it, it doesn't really go back to normal shape anymore, right? Okay, so that's what happens with our suspensory ligaments. Okay, once you stretch them, it takes a long time to go back to the original shape. So let's go back to here. When do you want to stretch it? You want to stretch it, you want to stretch the suspensory ligaments when you're looking at something far away. And you need the suspensory ligaments to return to the original unstretched state when you're looking at nearby object. But that's difficult when we're older because it lost the elasticity. And so when normally when just walking around, the normal state is like far away vision, right? But when you need to, look at something on your phone, or if you need to check the writing on the back of a bottle of your medication, for example, you don't see it clearly anymore. Okay, we call this presbyopia. Right? Presbyopia. Right? So that's why people need to move it further away to see, right? Or they need to have reading glasses. Right? It's because the suspensory ligaments, because of aging, it loses its elasticity, and they're not able to return to regular length quick enough. Right? So with presbyopia, you can't see the words on a nearby object. Okay. So here is the eyeball, and that's the lens. We have the anterior compartment here, anterior compartment, and we have the posterior compartment at the back. And I told you last time, the anterior compartment is filled with a nourishing fluid called the aqueous humor. And we need that because we don't have blood vessels in the cornea or in the lens. So the aqueous humor is how you would nourish them. And the back of the eyeball, we have the vitreous humor, right, which fills it up and it reinforces, right? Reinforce the shape. Sorry, reinforce the shape of the eye, and the aqueous humor nourishes the lens and cornea. All right. Last time I told you about floaters. If you didn't know what I was talking about, because you don't have it, and that's a good thing that you don't have it, but that's what it is. Okay, people with floaters, Like today is pretty bright outside. Like if I stare at the sky and I blink, then I see these debris kind of floating back and forth. That's what they are. Right? They are basically protein debris that have washed off from the lens or the cornea sometimes, and then they have nowhere to go. Okay? They're too big to be drained away, and then they just kind of stay there. If it's really, really severe, then there are drugs, eye drops, right, or laser treatment that could break down some of them. But for the most part, it's it's a nuisance. Okay, people living with these floaters. Cataracts is another eye condition. That is when the lens turns cloudy. Right? And eventually it will become completely opaque, and you won't be able to see anything because the light's not going to go through, right? But you can do surgery. You can replace the lens. Right? Nowadays they have artificial lens that you can put in anymore. You can put in now. Okay? You don't have to wait for like a donor to give you a lens anymore. Okay? You can use these artificial lens. My mom just had hers done. To on her left eye. She had cataracts on one, but not the other. Yeah, and now she actually after the surgery, she doesn't even need to wear glasses. Okay, because by replacing the lens, if you have nearsighted before, the new lens is going to correct that as well. Right? So yeah, and the government covers it. There is another one called glaucoma. Glaucoma is high pressure in the eye damages the optic nerve. Right? So your nerve is not communicating with the brain basically if it's damaged, so you won't be able to see anything basically, and it's a progressive loss of vision, starting with peripheral vision, and then it progresses over time. There's no treatment for glaucoma at the moment. There are only interventions that can be done to lower the eye pressure. So by lowering the eye pressure, you slow down the degradation of the optic nerve, and you slow the progression of the disease, basically. Okay, that's all. Okay, we're gonna take a note that explains the visual pathway to the brain. This is another confusing thing, and you're gonna need your study guide for this. There's a picture like this in the study guide. If you don't have it print out, I'll give you a moment to draw this thing out on the left here. Okay. Need it to be relatively big. Take up, like if this is your paper, you want to do this. And for this to make sense, you need to have at least two colors. Two colors would be good. All right, so what are we looking at? Right? This is if you look at the eyes from above. So this is your right eye, and this is your left eye. Looking at it from the top, okay? And that's gonna be like the pupil. Right? That's where the light's gonna go in. Okay. So things that are on the right side, on your right side, that's gonna be the right field of view. Okay. Right field of view, FOV. And we're gonna use red to represent that. Okay. So let's say, you have a person here on your right field of view. So to see the person, light has to bounce off from that person, right? If there's no light, you can't see anything. So light travels in a straight line. So to see the person on your right, the light has to enter the left half of your eyeballs. So this left half of the eyeball, both eyes, that's where the light from your right is going to enter. Do you guys understand that? The left halves of both eyes are responsible for seeing things on your right. It's a little bit weird, but let me know if you have questions. Okay. Think of like holding two mirrors, right? Like pretend these are mirrors. Okay. This mirror on the left will be collecting light from the right side, right? And this mirror on the right will be collecting light from the left side. Okay. I'm going to use a different color right here. This is the left field of view. All right, so like if you have another person here, it's going to go to the right side. The light bouncing off from the person is going to go to the right side. Okay, through the pupil, right? There we go. All right. Let's try to follow these information to the brain, and I'll tell you what we're looking at here. This thing here, that is the left optic nerve. It's a thing, the eyeball that we drew, there's always a thing that comes out at the back, right? That's the optic nerve right there. And then this is the right optic nerve. So let's follow the information from your right field of view. So from the optic nerve, from the left optic nerve, is going to continue to travel. It's going to go towards the middle of the body. Same thing over here. It's going to go. And then because they contain the same information, right, from the same side, they will join together in the middle, and then they will travel on the same side. Okay, let me try to do that again. They will travel to the same side like that. So the information from the left eye, will stay on the left side, but the information on the right eye will cross the middle and then go to the left side. Right? So this here is inside the brain now. Instead of the optic nerve, we now call it optic tract. You might remember nerve and tract are the same thing. They are a bundle of axons, but nerve is outside of the central nervous system, but tract is inside. the central nervous system. So that's going to take the information to the left thalamus, where it receives all the sensory information except for smell. It's going to filter it, and then it will send it to the left occipital lobe. All right. Now, a couple of things to pay attention to here. Both eyeballs will send information to the left brain, but the left brain only receives information about the right field of view. And let me say that one more time. Both eyes will send information to the left brain, but it will only be information related to the right field of view. Okay? Now let's trace the left field of view. Left field of view. So we have the information coming in from the left eyeball, right half of the left eyeball. It's going to cross the middle to the other side. But for the right eye, it will stay on the right side. It will meet with the other optic nerve from the left eye, and then they'll come together, and then they will both go. They will both travel. Like that. Okay, so this would be the right optic tract to the right thalamus, and then the right occipital lobe. Okay. So this location where the nerves from both eyes come together before separating again, this is called the optic chiasma. Optic chiasma. Now, if that sounds a little bit familiar to you, that's because, and I'll come back to this, don't worry, okay? That's because in lecture 6, we had a diagram which is right here. This one. Okay. This is the hypothalamus, the green thing here, hypothalamus. The purple is the pituitary gland, and right in front of it, that's the optic chiasma. There's a little blue thing here. That's where the nerve crosses in the midline before continuing. And that's the optic chiasma. All right. So there you go. That is very confusing, huh? So let's do a bunch of questions related to this and see if it make any sense to you. You know what? Why don't we go for a break, and maybe during the break you can try this out as well, or you can wait until afterwards. But we'll take it up when we return. Okay, at 9:45. So see you in a bit. Okay, let's take up these questions. Okay, first one. The right optic nerve carries information from which field of view? So it's quite easy if you have the diagram. And this is much harder if you have to do it from memory, right? But right optic nerve is here. And what kind of information is it carrying? Well, I see the red as well as the blue. Red is the right field and blue is the left field. So the correct answer is both fields. Both FOV. Left and right. What about the left optic nerve? Take a look at the left optic nerve. Right here, also 2 colors. So it's also both field. Both FOV. However, once it passed the optic chiasma, once we go to the tract, then it's a different story. The right optic tract, which is right here, it only carries information from the left. It's only blue here, you see? And blue corresponds, oops, what did I do? Blue corresponds to the left field only. Left FOV. And of course, also for the left optic nerve, right? Similarly, is going to be just the right FOV. You just look at the color, basically. Okay, next question. The right optic tract, which is right here, carries information from which part of the eye? So if we follow the blue lines and we go back to the to each of the eyeball, you will see that it's coming from the right half of the eyeball. Both eyes. Right half. And as for the left optic tract, if you follow it all the way to the eye, it is going to be the left half. Okay. Any question about any of these? I know it's a lot to take in. Anything you want to ask to make this a little bit better? All right. So that's the professional drawing, as I always say. It's basically conveys the same information. Okay? So you can match this with what we've drawn, and it should make sense. Okay. So basically, like if you're looking at a flower or something, okay, flower. The flower is gonna end up on this side of the brain, right? So like you're gonna see the flower this side, but then the stem is gonna be over here. So basically, the whole flower gets flipped when it gets to the brain. That's another way of thinking about it. All right, I have to show you another note for eye problems. This one we have to probably open a new page. Okay, we'll just do this one. This is class notes, March 11th to 15th. Okay. So type of lens. Types of lens. When it comes to wearing eyeglasses, there are 2 types of lens. Okay, there is the this one. This is called a converging lens. Okay. Or a convex lens because it bulges out in the middle. And what convex lens do is they take light and then it focuses it at one single point like this. Right, so convex lens, they focus light to a single point. An example of a converging lens would be the magnifying glass. You can burn things with magnifying glass just by focusing the sunlight. And then there is another kind of lens that's called diverging lens. Which is concave lens. It's caved in the middle. And what this one does is it will take light ray and it will spread it out. Spread it out. Spread it out like so. Okay, that's just like the middle of the lens. So if you wear glasses, do you know which one you're wearing? Let's find out. So typically people have two kinds of eye problems, right? In terms of like, wearing glasses. There is a nearsightedness. Nearsightedness. Nearsightedness, the medical term, I suppose, that's called myopia. Anybody have excuse me, anybody have nearsightedness here? Can you describe to us what can you see and what you cannot see? Anyone? No? That's okay. I have nearsightedness, and nearsightedness means I cannot see far. Cannot see far away clearly. And the reason is because the eyeball is too long. Right? Actually, you know what? I think I have to tell you what happens normally first. Sorry about that. Okay, so let's do a normally. Okay, normal. This is your eyeball. You have your optic nerve. And let's put the lens right here. Okay, so like you will have your ligaments and all that stuff, okay? But that's the lens. Oh, I just killed the eyeball. Let's put it back right here. Okay, so this is the lens, okay? Lens. And this is gonna be the fovea. Fovea, where you produce the sharpest image, right? So your natural lens in the eye is also a converging lens. It's also a converging lens. So this is what normally happens, okay? The light's gonna come in, and then your lens is gonna bend the light, and it's gonna be right on the fovea. Okay, so it focuses on the fovea. The lens focuses light light rays onto the fovea to produce a sharp image. There we go. So let's see what happens when it's nearsightedness. The eyeball is too long. Now this is an exaggeration, but I'm just drawing it in an exaggerated way to help you remember, okay? Like nobody's eyeball should be this shape, but you know, exaggeration, like I said. This is the lens. We have the optic nerve here. So look, the light ray comes in and it's gonna focus just like before. But because the eyeball is longer now, it's like moving the screen further back, right? So the image produced by the lens is in front of the retina. That's why it's blurry. It's in front of the retina now. So I want to ask the class, what kind of lens do we need to correct this condition? Should we be getting a converging lens or should we be getting a diverging lens? Look, the light ray is coming together and is focusing in front of the retina. What do you think is appropriate? Converging lens that cause light to come together, or diverging lens that spreads them out?
I think because I'm nearsighted too, I just realized it. And I'm looking at my glasses, and I think it's a diverging lens.
It is a diverging lens. Very good. Okay, so we're gonna put on a diverging lens. Let's take a look at how that's gonna help us. Okay, this is a diverging lens. Okay, I have to use different colors to show you, you know, what happens if you do have the lens. If you have the diverging lens, the light ray will come in, but now the glasses is gonna spread them out a little bit for you, so that when your lens focuses again, right, it will be right back onto the retina. Okay? So you need to spread them out just by the right amount to correct for the elongation of the eyeball. Right? The problem with nearsightedness is the light rays are coming together too soon. That's why it's in front of the retina. So by wearing the diverging lens, it spreads them out, spreads the light out just the right amount, so that when you converge them again, it will fall right back onto the retina. Okay, so you need to have a diverging lens. And how much you spread them out depends on how long your eyeball is. Okay? So there, nearsightedness.
I have a quick question.
Yep.
So like when I was like younger, I didn't have glasses, and like I think a year or two ago I got them. So did my eye just like randomly like get longer, or how did that work?
Yeah, sometimes it happens earlier on, sometimes it happens later on in life. There's no particular reason to. I mean, like people say, generally speaking though, if you don't need to wear glasses before the age of 12, then most likely you're not gonna have to wear it later on. But sometimes it happens. Okay, like it could just be like we're straining our eyes too much because we look at a lot of screens these days, right? So that could put strain on the eye, and it could change the, shape of the eyeball, and then maybe have to wear it. But typically, if you're able to make it through 12, 13 years old, you're not gonna have to wear glasses. And even if you have to wear glasses, it's not gonna be really bad eyesight. Okay, so I assume like you have very mild cases, right? Like even without your glasses, you should still be able to see relatively okay. Yeah. Yeah. Whereas for me, like if I take off my glasses now, I can't even see what's on the computer screen. That's how bad my eyesight are. Okay. So, anyways, there's a lot more I can tell you guys, but not enough time. So let's move on to the other thing, which is farsightedness. Okay. Hyperopia. Hyperopia. So for people with farsightedness, cannot see nearby clearly. They can see far away, but when it comes to close by stuff, they can't see it. Right? My daughter is farsightedness. Apparently, it's something that kids can be born with. So at the beginning, she was not like she wasn't reading as early as her older sister. We don't know why. We thought she was just like, some kids are slower than others, so we thought that she was just slower. But it turns out she had farsightedness. So like when we're reading to her and stuff, she literally can't see the words clearly. That's why she wasn't able to learn how to read. And then we found out she has farsightedness from the eye doctor, and then they gave her glasses. And now she can read. Okay, so cannot see nearby clearly, and in this case, the eyeball is too short. Okay. Again, exaggeration. Oh, that was a perfect circle. This is exaggeration, but too short. And then here's the lens. And the light comes in. All right. And then it's, let me try that again. The light comes in. And then it focuses the light behind the retina. The image is behind the retina. Okay. So in other words, the light rays are not coming together soon enough, right? Because now the screen is closer, right? So for these kind of eye problem, you need to have a converging lens. Okay? So the converging lens will take the light ray, it will cause them to come together a little bit, okay? And then your lens is gonna focus it just the right amount. Okay? So you need converging lens. What did I say is an example of converging lens again? Do you remember?
Magnifying glass.
That's right. So people with converging lens glasses, people who have farsightedness, you can tell because their eyes look bigger when they wear the glasses. Okay? Because it's a magnifying glass. Right? So let me show you a picture of that. This is I guess it's a model. He's wearing converging lens in one picture and diverging lens on the other. Can you tell which one is the diverging lens? Which one is the one that looks like a magnifying glass? Is it A or is it B?
B.
B, that's right. You can see the eyes are bigger, right? Okay, and that's because, like I said, it's magnifying glass. Okay, and another way you can tell is if you look at the person who's wearing the glasses from like an angle from the side, people with nearsightedness, because they're wearing the I might have actually I might have said it backwards. Okay, let me just clarify for you. Okay. This here, if the face is indented like that, then this is a person with nearsightedness. Nearsightedness. Okay, and he is actually wearing a diverging lens. Diverging lens, not the magnifying glass. Okay, I think I said it backwards earlier. When I was asking you in this slide, in the notes is correct. Okay, like, but when I was asking you which one is like the magnifying glass, I think I said it wrong. This one, when the face is bulges out like that, and you can see the eyes are bigger in this one, this is the one with the farsightedness. Farsightedness. And that's the one with the converging lens. The magnifying glass. Okay? So if you don't know if someone is farsighted or nearsighted, you can tell, right, just by seeing how it is. And I have a bit of a thing when it comes to watching a movie and someone's wearing glasses. I always like to comment on whether or not the glasses are real. Because if you look at the glasses and the person turns to the side and the face is like perfectly aligned, like as if there's no transition. You see how the face is indented here, right? And the face is bulged out here. If you look at the actor and then the face is like completely smooth in the lens, then the lens are not real. Okay, it's like, oh, those are just fake glasses, yeah, to fit the character or something. Okay. So farsighted, nearsightedness. For people who wear glasses, what's another reason why you need to have glasses? We talk about three things, and we need reading glasses for presbyopia. That's when you get older. You can't read the bottles anymore. You can't read the book. You need reading glasses. We have farsighted and nearsightedness. What's the fourth reason why you need glasses, huh? Anybody?
Both of them at the same time?
Well, if it's both, they actually cancel each other out, right? So like my daughter, the eye doctor is telling us when she grows up, the nearsightedness might kick in and it might cancel out the farsightedness. So she might not need glasses anymore. But what's another one? Well,
Astigmat?
that's right. That's the one. Okay? So this is astigmatism. Okay. Astigmatism, light scatter in a different direction because the lens becomes uneven. So at night is really hard for me to drive because I have really bad. Not only do I have bad, myopia, I also have very bad astigmatism, and yeah, and that is causing me to have difficulty driving at night because that's what it looks like to me. The light is scattered, especially when it's raining or snowing. The wet road makes the light scatter even more. I literally can't drive at night. It's very dangerous. Okay, so I let my wife drive instead. But yeah, you also have to wear glasses to correct that. Okay, so there you have it. I think we need to move on to the ears. So I'm just gonna give you the answers to these questions. You can look at it later. Okay. Something is a common vision condition in which you can see distant objects clearly. You can see far away, but nearby object might be blurry. Right? This one is hyperopia. This hyperopia is farsightedness. The name tells you what you can see. Farsightedness, you can see far. Nearsightedness, you can see near. Increase in eye pressure could slowly degenerate the optic nerve, resulting in glaucoma. Okay. Cataracts is when your lens becomes white. Floater is those things that are floating in the aqueous humor. Presbyopia is when you age, your eyes cannot accommodate anymore. Correct answer is glaucoma. Which of the following is an example of presbyopia? I just told you. You need to hold a book far away to read it clearly. Okay. Can't see because, the suspensory ligaments have lost their elasticity. You stretch it too much throughout your life. It's not going back to normal shape quick enough. Okay, this one I want to do though. Take out that picture. See if you can answer it. Go, go, go. Okay, I'm waiting for maybe more. Okay. So, damage to the right optic tract. You know, you gotta have this picture, right, with you on the test. It's gonna help you. You guys did pretty good on the quiz though, right? I am assuming you had your graph with you, right? That actually helped you with a lot of the questions. And I mean, if you didn't have the graph and still do well, congratulations. But oops, I'm in the wrong lecture. I can't talk and do things at the same time these days. But like for this lecture, right? You want to have this eye thing with you, this diagram. Okay, so the question is, if you damage your right optic, what was it? I forgot already. Right optic track. Okay, so right here, right optic track is right here. All right, it's all blue, and it's all from the left field. So let's see which one is the right answer. Visual information from the left field. So basically, you would lose half of the vision. Like you won't be able to pick up things on the left. Like you physically have to turn your head to see the stuff on your left, right? You know what I mean? So that's that. Done. So are we ready for more? Yeah, so next time you go to the eye doctor, right now you know a lot more about how the eyes work and stuff like that, right? They usually give you a printout for your prescription. Okay, if it's plus, if the numbers are plus, you know the prescription you take to the what do you call those? The glass, the places that sell glasses. I don't know what you call them. Glasses places. Okay, if the numbers are plus, it means you have farsightedness. If it's negative, then you have nearsightedness. Okay, and then that last number, you have three numbers. Okay. Last number is going to be for your astigmatism, if you have any. All right. Now we're going to move on to hearing. Hearing. So the ear actually has two sensory functions. Sometimes you can have both, like in the same eye. Korea, is that what you mean? Both what? Both far and near on the same eye? Really? I have never heard of that before. Is that something that you personally have, or you heard about it? Interesting. Okay, maybe I'll take a closer look at that. But based on my understanding, is they generally cancel each other out. Um, but I know sometimes. Oh, that's why you have two glasses. I thought people have two glasses because one glasses is for farsighted or nearsighted, and then the other glasses is for reading glasses. Okay. Um, but because reading glasses has to do with the presbyopia, right, which is a different thing than nearsighted and farsightedness. Yeah, I think that's what it is. Um, Again, right, because the longer eyeball causes nearsightedness, and the shorter eyeball called farsightedness. So it should cancel each other out, right? And then the two glasses are again for presbyopia and for whatever eye problem that you have. Some people have like bifocal, right? Okay, so like when you look down, the reading glasses is at the bottom, and then when you look up, that's for like nearsightedness. Yeah. Okay, so the ear. The ear has two sensory functions. Most people don't think about it. Okay, most people think the ear is for, obviously the ear is for like hearing things, but actually the ear is for detecting equilibrium as well. Okay, equilibrium as in like maintaining body balance. Just a quick quiz here. Which part of the brain is responsible for detecting equilibrium? Maintaining equilibrium, I should say. Help me out here from last class. Which part of the brain maintains balance and equilibrium? Come on, you can do it. No? Oh boy, a little bit heartbroken there.
The temporal.
The temporal, that's good try. Temporal is for listening, for processing auditory information. How about I jog your memory a little bit here? Do you see anything here that helps you maintain balance? And it's the cerebellum, that's right, okay? The little brain at the back. Right? That's three things for you. Posture, maintaining posture and balance, right? Store motor memory, as well as help you execute smooth movements. Okay, so the equilibrium aspect is gonna go to the cerebellum. And Patricia's right about the temporal lobe. It's where sound is. And I'll put all these together for you in terms of, what goes where. I have a summary diagram for you. We probably won't have time today. We will get to it next class. Okay. So the kind of sensory receptor in the ear, they are called hair cells. Hair cells. And it's a type of mechanoreceptor. Remember, mechanoreceptors can be stimulated by changes in pressure as well as body movements. Right? So they are called hair cells not because they have hairs on them, but because the cell membrane form these structures that looks like hair. And when they're stimulated, the potassium is going to come in and it's going to cause depolarization, and then calcium is going to come in and then it's going to release the neurotransmitter. Okay, so it's similar to the action potential that we learned previously. Okay, so we're gonna go and label the ear together. Please bring out the ear diagram. Sorry, my kids are crying. I'm just gonna go see what happens to them. Okay, I'll be right back. Please go choose this. Find this picture. Sorry. Okay, sorry. Okay, so there's this diagram right here. There is the ear, okay, but the ear has a special name. Okay, there's your ear. The ear is called the pinna. Okay, pinna. Okay, that's just the external ear, the visible part of the ear, and it helps collect sound from like a wide angle, so that you can hear it. And at the bottom of the pinna, you have the earlobe. Okay, the earlobe can be attached or detached, depending on your genetic makeup. Okay, like some people have it like dangling like this, and then other people, like if this is the face, right? This is the face. Some people have the earlobe dangling, and other people's have the earlobe like attached. Right? So that's genetically determined. And over here, we have this. Okay. That is the auditory canal or ear canal. Auditory canal. Okay. Or ear canal. And it basically helps funnel the sound towards the eardrum. And that's what this line is, the F. F here is pointing at the eardrum, okay, which we're gonna color green here. Now, by now, you should know that they never use the simple names, right? So even though it's an eardrum, but it's called tympanic membrane. Tympanic membrane, which is the eardrum. And the function of the tympanic membrane is to convert sound energy into movements. It's basically the reverse of a speaker. If you have a speaker, the speaker moves and that creates sound. But the eardrum is opposite. It takes the sound and changes it into movements. You know, people can take the temperature through the ear now. If you want to check it, if you have a fever or something, the thing that is sticking in the ear, they basically is checking the temperature of your tympanic membrane of the eardrum. So on the inside of the eardrum, on the medial side of the eardrum, it is connected to 3 bones. And here are the 3 bones. Oh boy, hold on. Here are the 3 bones. 1, 2, 3. These are called ossicles. Ossicles. And the ossicles, the 3 bones, they form a lever system that amplifies sound. It makes the sound louder, so you can transmit it, you know, further deeper into your head. Now this over here, running out of colors. This over here, that's just part of your temporal bone. Yeah, I know we don't have a label here. This is the temporal bone, which is outside of your temporal lobe, basically. Okay, it forms your ear canal as well. And there's a tube here. This one is called the Eustachian tube. Okay, or the auditory tube. Don't confuse the auditory tube with the auditory canal. Canal is like the ear canal. So like it's probably better to know it as the Eustachian tube. And what it does is it connects the nasopharynx to the ear. And it helps equalize pressure. With the surrounding. Now, normally the Eustachian tube is closed, but when you swallow, you open up the tube. Right, so when you swallow, the tube opens and it lets the extra pressure comes out from your head. When we go on an airplane, or if you get in an elevator, even sometimes, okay, if it's a really tall building, as we go up higher into the altitude, the pressure of the environment drops, right? And so you feel your ear gets plugged. That's from the pressure in your head is higher than the outside. So when you swallow, swallowing opens up the tube. Those extra pressure that was building up in your head escape through the ear, and then you feel fine. And then later on it gets plugged again, and then you will have to swallow and do that. People say chewing gum helps, right? It's not the act of chewing gum that helps. Is that chewing gum makes you salivate more and you swallow more, and then the swallowing is what's opening up the tube. Okay. So the ear is actually connected to the nasopharynx, the back of the throat, if you remember. Okay. So in movies, again, if you watch a lot of movies, you might know. In movies, sometimes people smoke and then they can blow smoke out of the air, right? And then one time I told this to the class, and someone said, "Yeah, I can do that too." And he's like, well, "Did you want me to demonstrate?" I'm like, "No, I believe you." But it's true, okay? Because it's all connected. That's why the smoke can come out. All right, further in, we have this snail shell looking thing right here. And that's connected to a bunch of weird looking tubes. And that's the inner ear. Inner ear. Okay. Everything that's outside here, that's the outer ear. And the middle ear. Middle ear is like, eardrum. The ossicles, as well as the Eustachian tube. Any questions about the diagram here? Okay. So what we will do next is we will zoom in into the middle and the inner ear and see what they do. So here's a zoomed in version. Okay, I'll try to use the similar color here. There is the tympanic membrane. Okay. Tympanic membrane, which converts sound energy into movements. If you're hearing my voice right now, it's because the sound that's coming out of your speaker is entering your ear, causing your tympanic membrane to vibrate. Okay? It's basically change in pressure that causes it to vibrate. That's why too much pressure can pop the eardrum and you can't hear anymore. On the medial side of the membrane, there are 3 bones. Okay? The 3 bones forms the ossicles, right? So that's the first one. It's attached to the tympanic membrane. And then it makes a connection with a second one right here. And then there is a third one. Right? So they go by collectively, they're called ossicles. We labeled it on the previous diagram, but each one has their name as well. The first one is called the malleus or the hammer. So malleus is more like the anatomical term, and hammer is like, oh, that totally looks like a hammer. That's what they say. The next one is called the incus. Incus. And it looks like an anvil. Okay, looks like an anvil. Let me show you a picture of an anvil. That is an anvil. Okay, I think the blacksmith used that. And the last one here is called the stapes. It's C, okay? C is the stapes. I know it looks like it's pronounced stape, but it's actually stapes. And the other name is the stirrup. Anybody know what a stirrup is? Like, what does this thing look like to you? What does this thing look like to you? It's a thing that you put your feet on, right? When you're riding a horse, or like if you're giving birth, like the stirrup. So that's what it is. So malleus, incus, and stapes, or you can say hammer, anvil, and stirrup. I honestly don't know which one are easier, but there you have it. Now, the stape is actually pressing on kind of a membrane. Right, so I gotta choose a color here. There is gonna be a membrane right here. It's covering it, so you can't really see it. Okay, but that's what it is. It's called the oval window. Oval window. I'll explain what that does later on. Okay, just know that it's covering it. Oval window. All right, what's this again? Anybody remember from last page? Take a look. What does it do? That's for, yeah, go ahead, Patricia.
The auditory canal.
That's right. The tube though, not the canal. The canal is right here. Okay, this is the auditory canal. Right.
Okay.
Auditory canal or ear canal. That's where you stick the Q-tip in. This is the eustachian or auditory tube for equalizing the pressure, right? You swallow, it opens up. And then there is like a bunch of space here. A bunch of empty space. That's just the tympanic cavity. Okay, so everything I've labeled here, this is the middle ear. Middle ear. Right? You might have heard of people having a middle ear infection, right? It's very common in kids. That's like infection that's going on in some of these things. Any questions?
Yes. Why does the air make that waxy sort of
Oh yeah, that's part of the immune system. Yeah, the waxy coating basically makes it difficult for bacteria to colonize. Okay, and you know, I know some people like to remove the wax. Like I think like it's part of like a spa thing that some people can do, right? Like, you know, they burn the candle, or whatever, and then they take the wax out. But people say, you know, you shouldn't really remove your earwax, because it's, you know, you're stripping the protective barrier, right? And that makes it more prone to infection. Yeah, but sometimes kids can have a lot of earwax, to the point that they have trouble hearing. And so sometimes they say pour like olive oil in and dissolve some of the wax and then let it come out. That's what my pediatrician told me to do for my kids. I don't know, depends, right? Some people say it's like a hygiene thing, like remove it, but you know, physiologically, it's a protective mechanism. Okay, inner ear, inner ear. So we have this snail shell looking thing in the inner ear. Let's just color that first. It's kind of cool to know that you have a snail shell in the ear inside. It actually does look like that 100%. So some people never really know that. That's called the cochlea. This is the part that is needed for hearing. Right? When people can't hear, then they have a cochlear implant, right? That's what this word is, cochlea. My grandmother is like 90 now. Like she has been losing her hearing for the better part of a decade. Okay, and then she refused to get a hearing aid. Finally, we convinced her to do it, right? And now she can finally hear. Because before, when you talk to her, you need to be like really loud, right? And even then, she can't really hear you. But with the implant, right? She say everything is so loud, can't sleep at night. She need to take it off. But she used to being quiet. Right? She used to not being able to hear anything for 10 years. All of a sudden, now it's so loud with the cochlear implant. Okay, this thing right here. What is this? Do you remember? What is this thing that I'm coloring? Please tell me. Someone tell me. Tell me you're with me. No? Because at the end of your
Oval window
okay.
on the
No, yeah,
stair.
the oval window is this thing, yeah. But this would be the stapes or stirrup, right? And it's going to be pressing on the oval window, which was not obvious before, right? Okay. But now if you remove the stapes, you can see the oval window. I still have not told you what the oval window does. Okay, it's complicated. I'll explain later. Now there is another window. You have two windows in the ear. The oval window, and then there is the round window. Round window. Okay, now long story short, I will give you a better explanation later. So basically, again, when you're hearing things, the sound waves go into your ear canal. It causes your tympanic membrane, your eardrum, to vibrate, right? And the vibration of the eardrum is translated into movements of the ossicles, the bones. You know, the hammer, the anvil, and the stirrup, they all vibrate. And then the stapes, or the stirrup, which is the last ossicle, is going to be pressing on the oval window. So there is the oval window, the membrane, and then this is the stapes that's stuck to it. So as the ossicle vibrates, the stapes is like pressing into the oval window. So inside this whole thing, it's empty actually. There is a bunch of like tunnels in it, channels we say, and it's filled with fluids. So when the oval window gets compressed, it sends a shock wave into the fluid. So it goes like that. You know, this is the shock wave, and then depending on like the sound, it either goes really deep or it just gets really shallow. But ultimately, it turns around and then it comes back out, and then it will cause the round window to bulge out. Okay, so when you push in to the oval window, the round window bulges out, and then when it bounces back, this one goes back in. So the stape pushes in the oval window, shock wave goes into the snail shell, the cochlea, and then it causes the round window to bulge out, and then they kind of go in out of sync. So basically, the round window is there to relieve excess pressure inside cochlea. Okay, I'll explain this more later, but now I need you to understand, the sound wave causes the ossicle to move, which then pushes on the oval window, and then there's a shock wave that goes into the ossicle, goes into the cochlea, and then it's going to come back out. That excess pressure is going to come back out from the round window. Okay. We'll come back to this later. Okay, there is more stuff here in the middle. That is the thing in the middle here, this whole area. That's connecting the cochlea to the rest of these tubes. That is called a vestibule. Vestibule. And the function of the vestibule is to maintain it's to detect, we'll say detect linear or vertical, sorry, detect horizontal and vertical movement. So there are two parts in it. There's like a body here, and then there's another body here. The top one is called the utricle, and the bottom is called the saccule. And the utricle detects horizontal acceleration. Like if you're in a car and it speeds up or slow down, like if you brake all of a sudden, you feel that braking motion. That's the utricle. The saccule is for vertical acceleration. So like if you're falling, when you're on a roller coaster and you go down the hill, that falling sensation that you're feeling, that's the saccule. Okay, so that's like the car moving, that's utricle. And then if you're on a roller coaster and you're going down, that's the saccule. Okay? Hope that makes sense. Okay, just one last thing here. I think we're kind of running out of time. There are 3 tubes here. Let's just color them differently. We have a tube like that. Okay, that's this one. We have one at the back. And we have one on the side. These are semicircular canals. Semicircular canals. So if you think of the corner of a room, it's like the corner of the room, you can look at that. The orange one is going to be parallel to the back, the wall behind me. That's going to be parallel to the orange tube. The yellow one is going to be parallel to the wall on the side. And then the green one is going to be parallel to the ground. Right? So let's label them first, and I'll tell you what they do. This one, the yellow one, that's the anterior. Anterior semicircular canal. Okay, I don't have space to fit all of it in. That's a full name. Anterior semicircular canal. J, as you can guess, is posterior. And then lateral is the green one. So the semicircular canal is gonna detect rotation of the body or of the head. Detect rotation of the head. And you know, if you cannot rotate your body without rotating your head, right? So like when you're rotating the body, you know it's going to be detected by the semicircular canal as well. So there are fluid in these tubes, and when you move the head, rotate it, the fluid will move and it will stimulate the hair cells. So the anterior semicircular canal is, you know, it's like this plane, right? Parallel to this plane, right? To that wall. And so, you know, if you nod your head, right, then it's gonna be, you know, it's causing the fluid to rotate. And so the anterior semicircular canal is stimulated when you nod your head and say yes. That's probably the easiest way to remember it. What about the lateral canal, the green one? What kind of motion does your head have to do to stimulate it? Remember, there's fluid in there that could move back and forth when you nod your head, say yes. And what kind of motion will stimulate the green one? Anybody? How do you move your head? So this one is shaking your head side to side. Okay, when you say no. No, no more. I can't take it. Make it stop. Side to side. How about the posterior one? What do you do? How do you rotate your head to stimulate the orange one? Okay, you would tilt it to the side. Tilt it to the side. Okay, so side tilt. Tilt to the side. Okay, so those are the three semicircular canal. Okay, so I want to ask you, when you are doing this, think of like figure skater, okay? When the figure skater is doing this kind of, it's called pirouette. Which one gets stimulated? Is it anterior, posterior, or lateral? Pirouette. Rotating like this. Okay, which one gets stimulated? Help me out. Tell me.
Lateral.
The lateral, that's right. Okay. What if you are doing a cartwheel? You know, cartwheel? Cartwheel. Okay. You know, cartwheel, right? Which one gets stimulated?
Posterior.
That's right. Okay. And then what do you need to do to stimulate the yellow one? Like somersault, right? Okay. This kind of rotation. So that's what a semicircular canal is for. It is for detecting a rotation of the head and the body. This is for detecting of acceleration, horizontal, vertical, and then the actual hearing is from the cochlea. And there is a nerve that takes all this to the brain. Okay, there's a nerve right here that takes it to the brain. Is called the vestibulocochlear nerve. So the information from the equilibrium and the sound, they come together and they go up to the brain. Of course, the equilibrium stuff is going to go to the cerebellum, but the hearing stuff is going to go to the temporal lobe. All right, that's all we're gonna do today. I mean, if I keep going, I feel like, you know, nothing's gonna go in. So we'll go through the PowerPoint slide next time. I'll explain more about, you know, the turning around and then the windows and stuff like that. I think we have to move the test. I am just not sure whether I should move it to the 20th or the 25th. Do you guys have other tests coming up like that, like around this period? Math, chemistry, or whatever? Do you know? What is it, Tohib? I saw you unmute. Did you say something?
Yes, like no, okay. For me, on Thursdays, I used to have your test.
The test
Uh,
is next Thursday that you have?
No, I don't know. I don't know when
Oh,
my
you
next
don't know.
test.
Okay.
Yes.
Yeah, I think maybe we can move it to the 25th then. Do you guys have anything that week? Like, is it better for the 20th or 25th? I don't know.
25th is
Can you make
okay.
it 25th? Yeah, 25th. Okay, yeah, sure. Let's make it 25th then. But the catch is we still have to learn new things, right? Like, because we're nearing the end. Right? So like, yeah, make sure you
Is
guys.
our lab quiz available?
The lab quiz, yeah, I'll extend the lab quiz. The lab quiz is not available yet, but when we change the test to the 25th, I'll push the date for the quiz later. Okay, the lab quiz. All right. Okay, so I'll make an announcement. We will definitely finish the year. Hopefully, first half an hour on Wednesday. We'll start the digestive system, and whatever we don't finish, we'll finish next week. Okay, but we definitely have to start the renal system next week as well, and then I'll move the test to the 25th. We might have to have a lesson on the test day as well. I don't know. We'll see. Okay, but keep your calendars open. All right, that's it for now. Thank you, and I'll see you on Wednesday. Bye bye.